Welcome back. We are gonna now go through the stages of mitosis. I think I said the last video was video 3, but it was video 4. This is video 5. Um, should be another quicker one. Just gonna quickly go through stages of mitosis, and that's about it. The next video will be all about the regulation. So, hopefully these stages are review and you've learned about them before. Just in case though, let's go through. So, kind of as we enter into mitosis and exit interphase and enter M phase, some things kind of happen at the same time. The boundary is kind of gray in reality, but think of it as early prophase, end of interphase. This is when our chromatin is going to condense, so it's going to go from the bowl of spaghetti to the X-shaped sister chromatids. At the same time, our nucleus is actually going to disappear, the nuclear envelope, the thing surrounding it and protecting it, as well as a mitotic spindle forming. Spindles are just like little grabby arms that are going to be the things that pull the centromeres apart. You can think of them almost like a steel cable that you just attach that we're going to wind in. And so yeah, we're just condensing, nucleus disappearing, and we're getting ready to move those chromosomes around. Then we head into prometaphase, which is a bad name because it is the phase that is in between prophase and metaphase. But prometaphase, our chromosomes are going to continue to condense, and they're actually going to attach to the spindle fibers, so that's some elements of prophase in there. But then they're actually going to start to move towards the metaphase plate, which is just like the center line dividing the cell in two. So initial starts of movement, again, in reality, the lines between these phases are kind of gray or thin. I don't know, bad metaphor. But this is really just kind of wrapping up what was happening in prophase with the condensation of the chromosomes attaching to spindles. The new thing is that they're moving. So metaphase, I think of this one as middle phase. They line up in the middle. Wow. They line up along the metaphase plate. They're all attached to their spindle fibers. They're getting ready to be pulled apart. So each chromosome is lined up, all in humans B46 of them. And each sister chromatid is ready to be separated. Then we get to anaphase. This is the movement phase. This one, sister chromatids actually separate. So we actually separate the two chromatids and we pull them to opposite sides of the cell, where they will be on their own for the next stage. So they're separated, they're on opposite ends of the cell, and now we get the final stage, which is telophase and cytokinesis. So this is where the actual splitting happens. Cytokinesis is like the splitting of the cytoplasm. So we're splitting the cytoplasm between the two cells. Our nucleus is going to reform and protect that DNA before it starts doing any major copying, growth, or anything like that. You don't want your DNA to get damaged. So your nucleus is going to form. Then you're going to get what's called a cleavage furrow, which is really the cell's just kind of pinching inward, and it's going to pinch off. And it's kind of like if you see a bubble forming into two bubbles, but it forms a nice peach emoji style shape here. And then they finally split into two genetically identical daughter cells, and mitosis is complete. So that's about it. There's not too much going on. We're going to briefly talk about prokaryotic cell division, but you do need to know how to draw these, how to recognize them from drawings, things like that. Prokaryotic cell division is a little bit different. When they reproduce, we call it binary fission. It's just kind of asexual reproduction by mitosis. But in this, chromosomes are going to replicate just like we do. The daughter chromosomes are going to move apart, and the plasma membrane is going to pinch inward and divide the cell in two. The main difference is that they largely have circular chromosomes, so the whole process kind of looks different. We're just kind of separating the two bowls of spaghetti into two smaller bowls of spaghetti. But in general, the same basic steps are there. We don't have to get rid of or reform a nucleus because there is no nucleus. Things like that aren't there, but ultimately it's just replicate the DNA, move the DNA apart, pinch the cytoplasm, split into two. Okay, last video, unless I decide to combine the last two for some reason, is going to be cell cycle regulation, which is some really interesting stuff. But that's next video. I'll see you there. Bye.